So next up, I'd like to introduce Crystal Gigante from the CDC, who will be talking to us about the current situation in the United States and what she's found in terms of US sequences. So Crystal, you should be able to share your screen now. Perfect.
All right, you can see the slides fine?
Everything looks good.
Great. So today I'm just going to provide an update of what we've seen in the US so far using monkeypox virus sequencing. I'll go quickly through the first few slides just because others have covered it a bit. I just wanted to highlight uh, the largest outbreak of monkeypox in Western Africa occurred in Nigeria in 2017 after decades of no identified cases. And then during 2018 to 2021, there were 8 cases imported from Nigeria to non-endemic countries. Since the reemergence of monkeypox in 2017 in Nigeria, there had only been 2 published reported events of person-to-person transmission outside of Africa. And in May of 2022, we all know this pattern of cases in travelers from Nigeria shifted, with many countries reporting monkeypox among persons with no travel history to monkeypox endemic countries. All of the available genomes from the outbreak thus far have belonged to the clade that was formerly called West African, now called clade 2B. Genomes published during the 2022 outbreaks all share a common ancestor with monkeypox virus from Nigeria. However, sequences from surrounding countries are somewhat limited. It's just a quick summary of the monkeypox genome structure, which we've already heard a bit about, but I just wanted to highlight the more conserved genes and essential genes mostly located towards the central portion of the genome, whereas less conserved genes across orthopox viruses and more genes involved in pathogenesis are located at the genomic termini, as shown in this picture, which has a single orthopox virus genome wrapped along 4 lines. So our approach currently at CDC is described on this slide. We use samples from a lesion swab where the total DNA is typically very low, and the monkeypox virus DNA is somewhere between like 0.1% to 5% of the total DNA in that sample. We do direct metagenomics shotgun sequencing using Illumina NovaSeq platform. We do some screening up front using clade 2 monkeypox virus PCR for most samples, based on CT value for that clade 2 specific PCR. And then afterwards, we do a cutoff of 35 average read depth to publish those genomes that are only high coverage. Exceptions to that include genomes with unique mutations or clinical significance that might be sequenced even if they don't, or published even if they don't fall into those categories. I have here the assembly methods we've been using, de novo assembly, which I'll talk a little bit about in a minute, and some long read sequencing, again, mostly just for cases of high interest with specific mutations. Just get rid of that. So 2022 US monkeypox virus sequences have fall into two lineages that were just identified in the previous talk, shown here in red and blue on this simplified tree. Lineage B1 contains the majority of US and global sequences from this outbreak, whereas lineage A2 contains 3 2022 US cases, a case from Texas in 2021, and has also been reported in India and Thailand. Most of those cases have had travel to either West Africa or the Middle East among the lineage A2. And of note, there's been less reported person-to-person transmission of lineage A2, and we're not currently sure why that lineage seems to be less observed or less reported. This is kind of a summary or rehashing of what was just presented, that we have seen, among others, we also saw increased mutations in the 2022 monkeypox viruses, which were many of which were GA to AA mutations, which is indicative of APOBEC3 activity, which is shown here in this graphic for the different lineages of monkeypox virus with clade 1 and clade 2A, which do not include the current outbreak. Most of the mutations shown here with a horizontal line representing a monkeypox genome and each bar representing a mutation, either APOBEC like mutations in blue and cyan or non-APOBEC in red or gray. And you can see clade 1, which was formerly the Congo Basin clade, most mutations are, or many are red and gray, but in the current outbreak and lineage A of West African or clade 2B monkeypox, the majority of these mutations are APOBEC or blue, shown here. So yes, that was one of the major differences in the mutations that we've seen in this outbreak so far. Another thing I wanted to bring up in this talk is we've been noticing several cases of large deletions and genomic rearrangements in about 3% of monkeypox virus genomes that we're sequencing. And these deletions are concerning about 500 base pairs to up to 20 to 30 kb, really large genomic rearrangements that have the potential to impact diagnosis or efficacy of therapies. These deletions have been reported, similar deletions in vaccinia, and there's a similar rearrangement to what we've been seeing reported for a clade 1 monkeypox virus in 2005, and that's been published. And these sorts of deletions have been seen before. There's a Bielsa monkeypox virus genome that contains a deletion in the terminal region as well. But we are seeing this in about 3% of monkeypox virus genomes in the US. And one thing we wanted to point out is the sensitivity of different diagnostic assays to these deletions. So any diagnostic assay targeting a gene within the terminal regions might be sensitive to this type of deletion, and that includes a monkeypox virus clade 2 or West African specific or monkeypox virus generic assays that were published, which target the TNF receptor within the ITR. So in summary, genomic surveillance thus far in this outbreak has revealed multiple lineages of monkeypox virus in the US, and monkeypox virus sequences since 2017 have exhibited evidence of host antiviral editing by APOBEC3. And we're seeing about 3% of monkeypox virus genomes contain large deletions or rearrangements. And we're hoping to have a preprint available describing those results in more detail this week, hopefully, but it's not out yet. Analyses are expected to continue as more sequences are reported. And there are way too many people to thank for all of this work. These are just some of our partners involving these data from the US and then references. So thank you again for the invitation to present this and happy to answer any questions.
Thank you so much, Paula. I mean, Crystal, that was fantastic. We have several questions in the chat that I can either read out to you, or people can feel free to unmute themselves and ask if they prefer. So I will read out a couple of questions. Let's see, the first one here is, did your cases of A2 present with the genital rash as many clade 2B cases have? Did the A2 cases present with lesion formation that was staggered like many clade 2B cases, or were they synchronized in their emergence and scabbing? And that is from Leon Cayley.
Yeah, so we're collecting information for the US cases from the A2, but we don't have that data available yet. We're kind of still compiling it. One of those cases was just identified recently, and the sequence should be out this week. Because of travel history, the male-male sexual contact question, it can be a little bit tricky for those cases. So we have been looking for that information and hoping to share it once we have compiled it. But also keep in mind, we're talking about 3 cases from the US this year and 1 case from 2021 where we might not be able to interview that person anymore. So it is a little bit tricky, you know, to compare an average clinical presentation of 3 to such a big number on the other side.
Thank you for that. And then Lex, would you like to ask your question?
So you said you did some long read sequencing also, right? Did you see any differences between the genome that you recovered from long read compared to the Illumina short read?
No, not very many large differences. The repeat regions are very different because those short reads won't cross some of the repeat regions that it seems the long reads will. So there's at least five repeat regions that look different between the two methods. Otherwise, homopolymer differences, which are known problems for the long read ONT sequencing that we're using. Otherwise, the sequences are pretty identical. We have noticed a few SNPs, a few possible SNPs, usually in low coverage regions that we're following up on to see if they're, because they were also from different swabs, different lesions. So we'll follow up on those to confirm if they're real or just because poor data in whichever technology produced that low coverage.
In addition, there are some low complexity regions, correct? They are also poor performing, right?
Yes, yes.
So I think we're going to move on now. Crystal, there's quite a few more questions in the chat if you would like to answer those there, but